All right, good evening, ladies and gentlemen. We are going to talk about a process tonight that's called transcription. Now, previously, we have been talking about DNA. We've looked at DNA replication. We've looked at sort of the history of the discovery of the shape of DNA and how DNA works. We've even looked at biotechnologies. Tonight, we're going to start talking about how does DNA actually do what it's supposed to do. So what DNA does is, remember, it tells our bodies what proteins to build. And so the first thing that's going to happen is we're going to make a copy of the DNA because the DNA always stays safely in the nucleus. It never leaves the nucleus. So it's going to send messages out of the nucleus about what proteins it wants the cell to build. So those messages are called messenger RNA, and that's what we're going to learn about tonight is the messenger RNA. The mRNA leaves the nucleus and then travels to a ribosome where it tells the ribosome how to build a protein out of the different amino acids. So today we got to figure out how mRNA is made. So this is not where it was supposed to go. I don't know how that happened. There we go. That's what we're looking for. Okay, so first of all, what we have is sort of an overview image on the left here. And I'll just sort of talk you through the big picture and then we'll get into the details. So on the left, I see sort of a clear or white shape that looks like a guitar pick. That's RNA polymerase, very similar to DNA polymerase. It copies one side of the DNA that's called the template strand. It doesn't copy both sides of the DNA. It has a very specific spot called a promoter where it recognizes recognizes that this is the beginning where it's supposed to copy. So remember that you have like 20 to 25,000 genes in your DNA. How is RNA polymerase supposed to know where to start? There's very special signals in the DNA that tell RNA polymerase where to begin. So then as the DNA untwists and unzips, the RNA polymerase copies one side only, the template strand, and you can start seeing that red ribbon forming. That's the mRNA. And then when it's all done, the RNA polymerase leaves. It can be reused as just about everything in the cell can be recycled. So let's talk about the step number one, which is called initiation. How is it that the RNA polymerase knows where to begin and how to begin? So it says over here, RNA polymerase attaches to the promoter region in front of or upstream of a gene. So if the blue lines represent the DNA and it's double-stranded, I see that it is anti-parallel, so the 5' to 3' business. So the green area represents a special series of letters in the DNA that are called the promoter region. And the promoter tells the RNA polymerase that this is where you begin. So before the RNA polymerase comes on, there's a part of the promoter that is sometimes referred to as the Tata box. And that's a whole bunch of A's and T's sort of repeating, semi-repeating. And that Tata box is identified by one of these proteins that are called transcription factors. And the first transcription factor grabs onto the Tata box, and then the other transcription factors follow. That is a signal then to the RNA polymerase when the transcription factors attach on that now it needs to click on, and that's when we're going to actually begin this process of copying the template strand of the DNA. I will mention that in prokaryotes, RNA polymerase binds directly to the promoter. It doesn't need the transcription factors. And then just don't forget about the Tata box. I love that word, Tata. Tata. All right, moving on. Elongation, so making it longer. So this part's pretty similar to what's happening in DNA replication. Just like DNA polymerase copies one letter at a time, it's adding the opposite letter. So does RNA polymerase. Only RNA polymerase is only copying one side, the template strand. And anytime that there's an A in the DNA, instead of adding a T, it doesn't have T's in RNA, it adds a U. That's the one, well, two big differences. It copies one side and it has a U. Trying to see if there's anything else. The non-template strand or coding strand of DNA, that one's confusing to me. The template strand is the strand that gets copied, but the non-template strand is sometimes called the coding strand, and that's always confused me, those two names. It's synonyms of each other. We'll have the same sequence as the RNA transcript with thymines replaced by uracils. And if you look at the diagram, maybe you can see that. Let's see if you can see it right there. Here's the C, here's the C, here's the A, and instead of a T, there's a U. C, 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 C. So the non-template strand is the opposite of the template strand, and so is the RNA. It's the opposite of the template strand. So that's what they're showing you, is that they're similar. We don't need that there. Okay, next we want to move on to the termination. You don't really need to know much about termination. Transcript production continues until the end of the transcription unit is reached. There are messages in the DNA itself that say this is the end of the gene that's being transcribed. There are a couple mentions here about how prokaryotes terminate the RNA signal, and you really don't need to know these, so don't worry about row independent or row dependent. So we can move past that. I actually kind of like this little video. It's really, oh no, this isn't the one. Never mind. Pretend. Sorry, I always like to waste your time. You know, that's my goal. There's another video coming up in just a second that I would like to show you here. All right, so what happens next? There are many different kinds of RNA. The three types that you really need to know are mRNA, tRNA, and rRNA. mRNA is what we're talking about right now. It's called messenger RNA, but be aware that there's like 10 different types of RNAs that have been discovered or understood at this point. So mRNA is the messenger. It's going to carry information from the DNA out to the ribosome about what protein to build. The transfer RNA, I often refer to as the trucks because it starts with a TR. So they're the trucks that carry the amino acids to the ribosome and they drop the amino acids off. And then rRNA is what ribosomes are made of. Ribosomes are made basically of ribosomal RNA. And so that's why they're called rRNA. All right, and here's something important, very important. In prokaryotes, the mRNA, oops, I didn't mean for that to move. In prokaryotes, the mRNA transcript is immediately translated. So that means as soon as mRNA is made, then a prokaryote reads the mRNA and builds the protein. That is not at all true in eukaryotes. It turns out that in eukaryotes, our mRNA is really complicated and has to be processed. And that's what we're going to talk about next. So all kinds of things have to happen to the mRNA before it can leave the nucleus and then head to a ribosome to be translated. All right, so, and then this is just an image showing that. So here in the bacteria cell, the mRNA ready to go. You'll see that a little bit more clearly coming up in future notes. And then in the eukaryotic cell, we have our pre-mRNA, which is what it is when it's first formed. And then it goes through all these processing things, which we're going to talk about, and then it becomes the actual mRNA that leaves the nucleus. Okay, so what are these mRNA processing processes? So this only happens in eukaryotes, not in prokaryotes. The first two things that are nice and easy, there's two things that get added to both ends. So on one end, we add what's called the 5' cap. It's also called, I'm trying to think of the other names, it's called the GTP or the something cap. I can't remember all the names. But it's basically a nucleotide that's added to one end. It's very similar to a nucleotide. Nucleotides have only one phosphate. This has 3 phosphates. This is guanosine triphosphate, which we've heard of a little bit earlier in our cell communication unit. So that gets added to the front, and it is added to the front end of the mRNA to help the mRNA escape the nucleus and make it through the nuclear pores, so that the 5' cap helps the mRNA to get out of the nucleus. Then on the other end, we're adding something that's called the poly A tail. It's a whole bunch of adenosines, so lots and lots of A's added on, very repetitive, just a whole bunch of A's on the end. It turns out that the A tails can be different lengths. Why are they different lengths? So mRNA is a short-lived molecule. It's a molecule that's intended to be used a few times and then is intended to be recycled. So it turns out that when mRNA leaves the nucleus, there are these little eating machines that are called exonucleases, and their job is basically to recycle products, but especially to recycle mRNAs. And so they basically, I make it sound like they're eating the mRNA. They kind of are eating the mRNA. What they're doing is pulling one nucleotide off the end of the mRNA at a time until they've recycled the entire mRNA. So why do we have short and long poly A tails? Some mRNAs are intended to last longer, and some are intended to have shorter lifespans. So they have shorter poly A tails, and the nucleases go through them, and that mRNA is recycled very quickly. If they have long poly A tails, it takes the exonucleases a longer time to recycle them, and so the mRNA can be used more times before it gets fully digested by the exonucleases. All right, and then once we've got the cap on the one end and the poly A tail on the end, then we need to deal with introns and exons, and this is called exon splicing. Eukaryotic genes contain large stretches of non-coding DNA. Sometimes people refer to it as nonsense DNA. That may or may not be a fair name for it. The name nonsense DNA implies that it's unimportant or useless, and it turns out that it's super important DNA. It just doesn't code for a protein, and so it's a part of the DNA that's just a little bit different. So the sections that are non-coding, that are sometimes referred to as nonsense, are called introns, and they are found in between the parts of the DNA that are important for coding our proteins, and those parts are called exons. So what we need to do in our nucleus is we remove all the introns. They get spliced out. That's the word. They get cut out, and that's where I was going to show you a video. So it says to produce a functional protein, the introns must be removed and the exons must be spliced together prior to the movement, of the mRNA transcript to the nucleus. This process is accomplished by a spliceosome, and that's what I was going to show you, a type of enzymatic RNA molecule. You really don't need to know anything more about spliceosomes, but I thought I would just show you because I like this little video in terms of imaging, sort of what does a spliceosome kind of look like. Give it just a second. It's like a 30-second video.
Once a gene has been located and transcribed into mRNA, it must first be edited before it can be translated into a protein. This editing process is called splicing.
So this
it involves
is the mRNA,
removing
the
non-coding
raw
regions
mRNA.
called introns, leaving only the protein-coding exons.
So the
In the
circle that
cell,
fell away
the
is an
introns are removed
intron.
by special enzymes which recognize specific sequences. These enzymes cut and rejoin the coding regions for translation into proteins.
Spliceosomes are doing that. Good. All right. Next, I hope it won't start another video like it always does, and then it drives me crazy when that happens. Oh, what I do? Give me a second. Next. One more. There we go. Okay, why are there introns? That is not entirely clear. We're still kind of solving why eukaryotic organisms have introns and prokaryotic organisms generally don't have them. Is it some kind of evolutionary baggage? Are they leftover genes that we don't use anymore? It's still something we're working on. However, what we do know, and this is super important that you understand, is that by having introns, it allows us to do something called alternative splicing, and this is wickedly important. What that means is when we cut out the introns, now we have maybe, let's pretend we have 3 exons. Those exons can be put together in 1, 2, 3 order, or in 1, 3, 2 order, or in 3, 2, 1 order, or in 2, 3, 1 order. They can be put in different orders because they've been separated, the introns have been removed. And so you can splice them together in different orders. And what that means is that you can have one gene, but you could produce a half dozen different proteins from that one gene. And that's called alternative splicing. And that is super important and gives eukaryotes all kinds of flexibility in terms of their the proteins that they can make. All right, ladies and gentlemen, I believe that is it. So have a good rest of your night.